Hi everyone, welcome to my biology skills exam. Today we're going to be starting with micropipetting technique. And today I'm going to explain and demonstrate how to use a micropipette. And for first thing, we got to calibrate 3 microliters. So we're going to do 0. Okay. Teague's over there. Hey Teague! Okay. So how you do 3 is it's going to be 030 because that's like the tons place or whatever. So, yeah, so we got 3 over here. For this one, we're going to do 0.26 because we're calibrating 26 microliters. Come here. It's going to take a while. I did a little high. And so that's 0.26, and the red line is on the 0 for the 6. It just makes sense. I don't know. And then 720. Here's what we're gonna do is so it is, and it's on the 0, so it's 0.720. There you go. And explain how you would convert 25 microliter, whatever that is, to microliters. You do like 0.25. So that's how you do it. Okay. And now for loading a gel electrophoresis. Explain why you use agarose gels and buffers for gel electrophoresis. It's to get like a positive negative charge and have the colors move from side to side. And agarose gels just like correspond with like the charge that you're giving in for it to move. And you're also able to see like which dye people use. And okay, just got the process of casting a gel and loading the gel in the chamber. I don't have a chamber. Casting a gel, well, it's a pretty easy process. You gotta calculate how much agarose powder you need based on the amount of buffer you use, and like if you use like 0.5 to every 100 meters. That's just how I do it. And now I'm going to. And then loading a gel in the chamber, you have to make sure everything's screwed on, screw it down and stuff, and then you pour it over. And you cannot move it because if you move it, all spills out from personal experience. Am I right? Am I right? And you're going to put it in plastic wrap. When it's all dry, and you're going to put it in the fridge. And then you're going to wait till like the next day, and then you're going to do it. Okay. So now I'm going to show you how to load a gel in the silicone practice gel. Oh my gosh, I'm gonna use 26. Oh, actually, we're gonna do 0.10. Hold on. That's that's like my lucky number for this one. So now we have 10. Let's get started. You're going to take a pipette tip and stick it hard. Don't touch it. And okay, we got some color. And okay, here we go. I'll like show you how to do this. Can you see that? We're gonna hope. Okay. So you're going to open up your dye. Very good. Place it right there. And now, here, I'll see you in the next one. So you're going to do a short stop, which is how you're going to get it. Make sure it touches all the way to the bottom. And then you're going to let it flow. And then pick it up. Have the dye. And go into the well. Just going to feel for it, and you're going to do a hard stop. Okay. So that kind of went, but it's in there still. It's in there, but it flew out, but that's sometimes what happens. It's just how it is. Okay, now I'm done with the pipette tip. So I don't have a garbage can, but we're just gonna go like that. It's important to add a loading dye for gel electrophoresis to see, because so what we tested was there real dye in like the Skittles that we took the dye from, and it was because the Skittle, because like the true like blue 5 I think it is, or like blue 20, I don't know, something, it would expand to the same place that the other blue would. It would just be more colorful, and I don't know what the word is, but it would just be more bare instead of the other one, because the other one is like diluted by like the sugars, and it's just it's not like pure dye, you know. So you can see if it's truly like the blue 5 or whatever it was. And where would you position the wells? Why? Explain your answer. From the very top, because they do expand quite a ways away until at least like the middle or even like the end of it, depending how much like loading dye you use and how much like microliters you use to load your dyes and stuff. So you want to place it at the very top, and then it can flow from the charges. And now Centrifuge Techniques. Draw on the whiteboard how you would place 3 micro centrifuge tubes. Or how about 6, 10? Okay, yeah, you can do that. So if there was 6, this marker does not work, I don't think. Okay, that's fine. We can work with that here. Sorry, ladies. Here we go. Oh yeah, by the way, if you are a female in this class, take it with the female friends. I'm the only female. Sucked. It was a really fun class, but I missed my other females. Okay. Okay, so we're gonna start with 6. 2, 3, 4, 5, 6. And how would you place 3 in this one? You would go. Okay, I need to think for a second. Okay. Here's how I would place 6. As you can see, they're even, and because everything has to balance each other out, so it doesn't such a huge like one over the other, you know. So everything has to be completely even, and that actually is even. It's just really weird distortion, I think. Yeah, that's even. Okay. Now for 10. Bigger circle. 2, 3, 4, 5. 1, 2, 3, 4, 5, and this is again how I would place 3 tubes, but in a 10, as you can see, they're all balanced out, and that's because everything needs to be balanced perfectly, so one doesn't over one doesn't over centrifuge the other, you know, so everything mixes evenly. And what would you do if you only had 1 tube to centrifuge? You would have to add another empty tube in, or just something, because it would not be balanced and it would ruin your sample. So you need another tube to do it. Explain the difference of pulsing and that of centrifuging for 10 minutes. Pulsing is where you just mix it a little bit, and it's not really that extreme. It's more of just like short mix to like separate the things or to combine them. And then centrifuging for 10 minutes, it fully like mixes it from each other. So it like fully separates it. Like there's nothing, this is just like if you just want a little bit, if you just need to get some out, or if you don't have that big of a sample. And now for streaking, place for bacterial studies. Explain what you need to do before and after any bacterial labs. You need to wipe down your lab station with bleach. So you spray it all over with bleach, and you take some wet wipes, or not wet wipes, but you take paper towels and you wipe them down. And draw on the whiteboard provided how you properly streak a bacterial sample on a plate, 2 points. Okay. We need to erase from my centrifuge journey. So, this is my little petri dish, and you're gonna start out with going on one side, and then to here, and then to here, and then to here again. I call this my little lucky number 1 through 4. That's how I start a bacteria plate. I go 1, 2, 3, 4, and you can go like more. It doesn't have to just be like this. Like you can go like, it can be wider, you can do much more. It depends on how much you have in your little thing right here. And I would just go like 1, 2, 3, 4, and when you go all around, you can get a lot of the bacterial like colonies that you want to see, and that's just kind of how it works, I guess. and explain why you need to streak that way. You need to streak that way so you can attain all the bacterial colonies that you need to get. And demonstrate how you would streak the plate. And actually, to streak it also correctly, you also have to go in the middle. And that's why I call it my lucky 5-star streak. Because then you end up in the middle, and that's a 5. I'm not going to write a 5, but it's just the middle of it. So it's like this big star. And that's how you finish it off. And it's demonstrate how you would streak the plate at asapictally. I know it means like sterile, but I don't know how to say it. So again, I'm just gonna show you guys this again. You're just gonna go down 2, 3, 4, 5, and you cannot let this touch anywhere else because or else it's going to ruin your sample. You're gonna have other bacteria that you don't want. You just want like your plain bacteria. So make sure you're very clean with this. If you touch it on the table, get a new one. It's just how it is. And experimental controls and design. Okay, we need to think about this for a bit. Explain what a positive control provides us. A positive control is like here. So a positive control can show, because you always want to see the opposite to make sure yours didn't turn out like with contamination or anything else. So a positive control provides us see like what like the positive outcome would look like, so we can compare it to our negative outcome and so forth. Moving on with the negative control, we can see what our positive outcome looks like, or we can see like if like we didn't think our positive outcome meant that much, but then the negative control was really much different. Like it's how we can compare it and create our judge and like our and we can create our opinions on like the lab notebook entries and stuff like that. And then explain what could have happened if your negative control yields a positive result. It's just accidental, like positive. That can be attained from like contamination and other stuff in your plates, and that's why you need to streak with a clean little swabber thing. That's what I call these swabber things. And it needs to be clean. It cannot touch the surface. Like if it touches the surfaces, then you put in your petri dish, it's just gonna like, it's not gonna go well. And then explain what happens if your positive control like yields a negative result. Again, with the contamination, you really need to be careful with this, and that's like the only way you could get it is from cross-contamination in lab industries. Also, washing your hands, wearing gloves, wiping down like your lab station. There's just a lot of stuff that goes into that. Presses are used to mix the tubes in a centrifuge and like samples evenly or like at high speed on power, and you can choose which one, and that's kind of like what it's used for. And lastly, Supernatant is the liquid lying above the sample that was separated during the centrifuge. So
am I talking about it for 10 minutes, or am I talking about it for 10 minutes?
I don't know. You have to answer that.
sorry, I thought I messed up all the questions. Here, wait. Hold on. And lysate is like the cell, like cells and like cell material that like comes from like the lysis of a cell, and it comes out during centrifuging. But then Supernatant is like the liquid above that. So usually would be done, but I'm gonna go back to number 11 to explain more because I don't think I explained that much on that. And the difference between pulsing helps when you have like a very low amount of sample in something, so low that actually it wouldn't even be like this much, but it would look more so just like the dots like right here instead of not this. So it would just look like this, and you would pulse it to make sure it all like collects at the bottom so you can micropipette it out easier. And centrifuging is like for 10 minutes is when it's completely separated, and you can separate the sample, and you can use like the Supernatant or like the lysis, and you can like empty out like it's not backwash, but like the something, and you empty that out, and you can use the Supernatant for like your other part in the labs. And that is my biology skills exam. Thank you so much for watching, Miss Manor. I really hope I get a good grade on this. Thank you.